Welcome to all in Biochemistry. In today's session, we will discuss about the DNA replication. In today's session, we will discuss about what is DNA replication, what are the main components of DNA replication, and replication process in eukaryotic as well as prokaryotic. Okay, so we start today's session and understand complete information about the DNA replication. Okay, so now first we understand what is a DNA replication. Okay, in DNA replication process, DNA plays a most important role. Okay, now in which form? This DNA is a genetic material. Okay, it will transfer the information from one generation to another generation. Okay, and it will also code some information. Which types of information? To code a protein or to synthesize a protein. Okay. What is genetic? What is DNA? DNA is a genetic material. It will code the information in a form of which form and which information? To synthesize a protein. Okay. During this protein synthesis, a series of process will happen. In this series of process is known as a central dogma. And what are the main steps of central dogma? First step is a replication. What do you mean by the replication? Replication means DNA will self-replicate. Means DNA will form their own copy. Okay. After this replication, DNA is converted into the mRNA. And this process is known as a transcription. Okay. And then finally this mRNA is converted into the protein. And the process by which mRNA is converted into the protein is known as a translation. Replication, transcription and translation is known as a central dogma of molecular biology. Okay. During this central dogma, first step is a replication. Okay. In these replications, DNA will self-amplify. Means DNA will amplify into the protein, to DNA. Okay, like this. And our DNA is a double helical structure. Antiparallel strand contain the two strand. First strand is known as a template strand. And second strand is known as a complementary strand. Okay. And the direction is 5' to 3', like this, 5' to 3', and 3' to 5'. Okay. And in a case of eukaryotics, DNA is tightly packed with the help of histone protein. Histone protein. During the amplification or during this replication, the modification will happen in the histone and replication process will start. The fundamental of replication. Now we discuss about the types of replication. DNA replication. Okay. DNA replication mainly semi-conservative. Maybe semi-conservative. Semi-conservative means during the amplification process, one strand is old and second strand is new. This is means first one strand is conserved and second is new one. This is old one and this is new one. Okay, this is the semi-conservative model. Conservative and one another model is a conservative. Conservative means one strand is conserved, second strand is new one. Okay, this is the conservative. Next is a dispersive. In dispersive means some part is conserved, some part is new. Conserved, new, conserved, new. Like this. And now this is the dispersive. Okay. These are the three models. Semi-conservative, one is conserved, one is new. In conservative, one is old and another is new. In dispersive, some part is conserved, some is new. Some conserved, some new. Some conserved, some new. Okay. DNA will generally semi-conservative and the mechanism is vary. It is depend upon the structures. Your DNA may be linear, may be circular, may be nuclear, may be mitochondrial, may be chromosome, may be plasmid, may be viral. Okay. Depend upon the types, location and structures, it is divided into different model. First is your theta replication or theta model. Okay. In this theta model, it seems in a double stranded DNA. And the best example is a E. coli or prokaryotic replication. Okay. Like this. DNA, circular DNA, amplification will start and form like this. One strand is old one, second is new one. Old, new, and it will amplify like in this direction. And this is in this direction. Okay. And it will convert it into the 2. This theta like structure is formed. That's why this is known as a theta replication. The production of theta replication during the 2 circular DNA. That's why this is known as a theta replication. Okay. And it will initiate with ORI region. Some helicase will bind. Then it is a bidirectional. Okay. Then same process. Then DNA polymerase will bind. Lagging strand will form. Logging strand will, leading strand, lagging strand will form, and DNA will replicate. Okay. And detail discuss. We will discuss and the detail about this detail procedure we will discuss in our incoming slides. Okay. A special feature is intermediate look like a Greek letter, that is the theta. and this is the theta. Okay. Importance: ensure the faithful duplication of DNA of bacterial chromosome before the binary fission. Okay. Next is a rolling circular DNA or rolling circular replication. In this model, first band will nick, matlab in one strand nick will form. Okay. One strand is breakdown, then new strand will form like this. Okay. One is old and new is released one. Okay. First cleavage at this point. Now this is the 5' end, this is the 3' end, and at this 3' OH group, DNA polymerase or DNA other replication machinery will bind and start the replication process. Okay, and this old one is removed out, then finally cleavage, and DNA will replicate. Okay, here no breakage, but in a rolling circular, one breakage, one DNA strand is break. Okay. New strand display old one, and old strands can become a double stranded based on a complementary. Okay. This is also a type of semi-conservative because here a one strand will conserve and one is removed out. This is also a type of semi-conservative because a one strand is conserved and second strand is new one. Okay. Now linear. This linear replication process is performed in eukaryotic chromosome. In linear replication, number of ORI regions are present. जैसे ORI region 1, ORI region 2, ORI region 3. Then here on this region, replication machinery will load. Then this replication fork will happen. Then proceed like this. Then new DNA will amplify, and this is also a type of semi-conservative because one is old and one is new. Okay, these are the three main mode of replication. First is your theta replication, second rolling, and third is linear. Okay, these. Now we understand the main step of replication process, both in eukaryotic as well as prokaryotic. This replication process is divided into three parts. First is your initiation, second is your elongation, and third is your termination. At this initiation, replication process will start. At this elongation, DNA will elongate, and at this termination, process will stop. Okay, this is the replication. These are the main three steps of DNA replication. At the initiation, first and most important component is the ORI. ORI at a region, ORI at the point where the replication process will start. DNA A, DNA B, DNA C, DNA G. These are the enzymes. Okay, it will help to form a replication fork. Then SSB, that is a single binding proteins. Okay, then CDC6 and MCM. Okay, these are in a case of prokaryotic, and this is for eukaryotic. These are the components which we require in initiation step. Okay, then in elongation, in DNA elongation, we require DNA polymerase, DNA ligase, and SSP, that is a single binding proteins. Okay. In termination step, we require telomerase enzyme in case of eukaryotic, TUS and TER protein in case of prokaryotics, and topoisomerase to remove out the positive and negative coiling. Okay. These are the most important component during the DNA replication process. Okay, now we discuss each and every point in detail. Okay, first ORI, that is the origin of replication. At this ORI, replication process will start. It will contain some specific reasons, specific sequences. Okay, and this sequences is AT rich. AT rich means adenine, AT rich means adenine thymine rich. Okay, ORI, origin of replication, contains some specific reason. It is a starting point. Okay, in case of chromosome or plasmid, this reason is known as a ORI, that is the origin of replication. Okay, and general feature, it is a AT rich. AT rich means it will contain the multiple AT base pairs. Okay, and adenine bind with thymine to double bond. Okay, and this adenine bind with double bond, and this double bond is easily breakable. That's why this reason is AT rich. Okay, sequences means a specific recognition motif where the initiator protein will bind. Okay, in case of prokaryotes, this contain a 13-mer or 19-mer sequences. Okay. Then binding site for the initiator proteins like DNA A in a case of prokaryotic, ORC in a case of eukaryotics. Okay, must ensure once per cycle replication to maintain the genomic stability. These are the specific, some general features of ORI. Okay. Now we discuss the difference between the ORI region of prokaryotic as well as eukaryotic. Okay. If we're talking about the number of genome, then in prokaryotics only one ORI region is present. But in case of prokaryotics, multiple ORI region is present. If we're talking about the size, the ORI region contains some specific sequence and then length is 245 base pair. But in case of eukaryotics, this is the 100 base pairs. Okay. Control by the DNA A protein or sequence A. And in eukaryotic, it will controlled by ORC complex and CDKS proteins. Okay. Replication timing in prokaryotics, it is continuous. In eukaryotics, as phase regulated. Okay. Sequence specificity, in case of prokaryotic, it is highly conserved, but in case of eukaryotic, it is flexible. Okay, and these are the some pointer, and these pointer are important for exam point of view. Like E. coli ORC region, it is a DNA box, it is a AT rich. It will contain 13-mer and 9-mer sequences. Okay. In case of eukaryotic, it will contain ARS region. Okay. This ARS region, ORC will bind and ACS sequences. In case of human, multiple ORI region, epigenetic regulation. ORI ensure the bi-directional replication fork. In plasmid ORI, call even commonly used for the cloning purpose. And these are the most important pointers for the exam point of view. Okay. Now, initiator proteins. Initiator proteins means initiator protein are the first protein that recognize the ORI region and help to start the DNA replication process. Okay, then unwinding the DNA locally and the recruitment of the other proteins like helicase, primase, DNA polymerase to form a pre-replicating complex. Okay, now we discuss in case of prokaryotics. In case of prokaryotics, the initiator protein is a DNA A. This DNA A is the initiator protein. Okay, and how it will helpful? First step is the recognition of ORC region. Okay, and this ORC region, the size is 245 base pair, and this is 80 wrist. It will contain 9-mer sequences, 9-mer or 9-mer or 13-mer means number of base pair is 13 at this region and number of nucleotide, number of base pair 9 at this region. And and this DNA A recognize the 9 base pairs, that is known as the 9-mer, and this is the sequences AT A T C C A C A. Okay, at this point DNA A will bind. This region is a AT rich, that's why it will easily unwind. Okay. Okay. Now at this position, the initiator protein DNA A bind with ORI region. Let's suppose this is the region. Okay. At this position, DNA A protein will bind and it will form. Unwind this region like this. Okay. Now oligomerization. Oligomerization means multiple DNA molecules form a complex around this ORI regions. Okay. Now at this position, DNA will tilt. Why it will tilt? Because here the torsional strain. Okay. At this position, this causes the DNA bend or introduce the torsional strain. This leading the local unbinding of AT rich region. Okay. Then further recruitment of helicase. Helicase means DNA B. DNA B is a helicase enzyme. Okay. Then this DNA A recruit the DNA C, and this DNA C helpful to recruit the DNA B. And this helicase enzyme is responsible to unwind the DNA. Now this unwound the region. Okay. And this reason, the ATP dependence means DNA A require the ATP for the functional property. This is the ATP dependent reactions. Okay. And the key initiator protein is a DNA A. In short, first DNA A recognize the ORC region. In ORC region, basically it will recognize the 9-mer sequences. Okay, and it will and for performing their action, it required the ATP. Whenever this ATP will hydrolyze, due to the hydrolysis, here the unwinding process will start. Okay, a number of DNA A protein will bind. It will create the torsional state. Due to this torsional state, this the hydrogen bond between the adenine and triphos in adenine and thymine, the hydrogen bond between the adenine and thymine is break down. Whenever this bond will break down, then DNA will unbound at this position. Whenever this DNA is unbound, then DNA A recruit the DNA C, then DNA C recruit the DNA B, and this DNA B is a type of helicase. Helicase is a process, helicase is an enzyme, it will helpful to unwind the DNA. Okay, this is the initiation process of prokaryotes, and at this initiation process, DNA A is a initiator protein, and it is the signature protein of this step. Okay. Now, initiator protein in eukaryotics. Okay. The eukaryotic replication process is more complex as compared to prokaryotics. Okay. In eukaryotics contain the multiple ORI region. It will contain multiple ORI region. Okay. And this regulation or this DNA replication process is compact and tightly regulated by the cell cycle. Basically in G1 phase or in S phase. Okay. In G1 phase, initiation will start, but in G1 phase, this initiator protein will inactive. At this S phase, it will convert into the active form, and replication process will start. Okay. G1 phase means preparation state, and S phase is a synthesis phase. Okay. Here ORC protein plays a most important role, ORC1 to ORC6. ORC1 and ORC region recognize the ARS region. ARS region is a, what is this? This is the origin of replication point, meant at this point replication process will start. Okay, and here this ORC protein is similar to the DNA A, and DNA A is an initiator protein of prokaryotes. Okay, and this ORC will bind at a G1 phase. Okay, after this it will recruit the CDC6 and CDT1. Okay, it will bind. Okay, and this is then MCM protein will bind. What is this MCM helicase? It is a type of helicase protein. It will helpful to unwind the DNA. MCM means mini chromosomal maintenance protein. Okay, in G1 phase, this MCM is in an inactive form. Okay, whenever the cell is go to the S phase, then kinase means CDKK or CD and DDK will bind. In G1 phase, this complex in a form of inactivate. But in S phase, some kinase protein will bind and convert it into the active form. Okay, and in S phase, initiation process will start. Okay, now what is the key differences? Key differences is that in prokaryotes, main initiator protein is a DNA A, here ORC regions. Here the DNA region recognition ORC. Here multiple. Helper protein is a CDC-C. Here the helper protein is CDC6 or CDT1. Helicase recruit, that is a DNA V helicase. Here MCM complex. Regulated by the ATP hydrolysis. It is regulated by the cell cycle kinase, that is the CDKK and DDK. It will helpful to activate the initiator complex. Number of region is only one, and in eukaryotes multiple. Okay, these are the key differences or comparison of initiators in eukaryotes as well as in prokaryotes. Now the exam point 2, DNA is a prokaryotic initiator protein, ORC is a eukaryotic initiator protein, DNA-C is the helper protein. In prokaryotes, CDC6 and CDT is a helper protein in case of eukaryotes. And in prokaryotes, this whole process is controlled by the ATP hydrolysis in case of protein. In case of prokaryotic cells or in case of eukaryotic cells, this complex is maintained by the cell cycle kinase, that is the CDKK and DDK. Okay. Now next is a helicase. This helicase is an enzyme. It will helpful to unwound the DNA. Okay. It will responsible to unwinding the DNA. Helicase is a ATP dependent molecular motor that move directionally along the nucleic acid strand to separate out the two strand while breaking the hydrogen bond between the base pairs. Okay, if we're talking about the types of enzyme, it is a motor protein and it is a type of ATPase because it will require the ATP to perform their task. Then energy sources is a hydrolysis of ATP in ADP. Okay, action, it will unwind the DNA, means it will break down the double stranded. It will convert the double stranded DNA into the single stranded by breaking the hydrogen bond between the nitrogenous phase. Okay, if we talking about the directionality, the directionality is 5' to 3' and 3' to 5'. Okay, now in case of prokaryotics, helicase protein is a DNA B. In case of eukaryotics, helicase protein is a MCM complex. Okay, if we're talking about the structure, in case of prokaryote, it is a hexameric protein. Okay, CDC-C and CDV-B helpful to load the DNA-B proteins. Okay, if we talking about the polarity, the move 5' to 3' on a lagging strand, but 3' to 5' on leading strand. Function, it will unwind. Okay. In case of eukaryotic structure, it is also a hexameric protein. CDC45 and GINS complex will helpful to recruit the MCM proteins. Okay. Polarity 3' to 5' in leading strand. Open to convert the double stranded DNA into the single stranded DNA. Okay. And also coordinate the DNA polymerase enzyme. Okay. Thumb pointer, prokaryotics, helicase 5' to 3' at lagging strand. MCM complex 3' to 5' activated by the CNG in leading strand. And processes depend upon the energy, basically ATP hydrolysis. It is a hexameric space structures. Helicase is a part of Replication. Okay. Mutation, it will form the genetic instability and diseases.